Hello, everybody. So we'll just wait for 1 minute for everybody to join and then we'll start the session. Okay. Okay. So let us start today's class. So a very good afternoon, everybody. So yesterday I couldn't take the class because I went for a conference. So I wasn't there. I was traveling, so I couldn't take yesterday's class. So that's why, like, I have already informed you, I think, in my previous class that I won't be able to take this week's class because I'm traveling. So, but anyways, today we'll do the uh, q- uh, study about lecture 11, and then we will have another class where we will do the questions of lecture 12, and then I think it would be over for this semester. So, I hope that you have studied a bit about the lecture 11, what all was there in lecture 11, and lecture 11 is basically about the telomeres. So, in the last lecture, we stopped at telomeres, and this class will continue about the telomeres and we'll study about telomeres only. So, a very fine discussion about telomeres we'll do. And this topic of telomeres is important. It is important because, let me just open it. Yes. So, this topic of telomere is important. It is important because uh, not only for your this exam point of view, but in general for your any competitive examination point of view, telomeres topic is important. It is a frequently asked topic, and it also is important for building your basic concepts related to molecular biology. Okay, so let us start today's class without any further delay. So week 11 is about telomere, telomerase and impact on genome. So telomere is different, telomerase is different. You should know the difference. Telomerase is the enzyme, okay? And telomeres are the repetitive sequences which are present at the end of the genome. Telomeres are added with the help of the enzyme telomerase. So telomerase is the enzyme which helps in the addition of telomere, and telomeres are the repetitive sequences which are present at the end. I hope it is clear to everybody. Okay, so let us see what is a telomere. A telomere is a region of repetitive DNA sequences. Let me just do it like this. Yes. So let us start today's class. First of all, let us try to understand what is actually a telomere. So a telomere is a region of repetitive DNA sequences at the end of a chromosome. Important here is that it is repetitive and it is present at the end of the chromosome. Okay. Telomeres protect the ends of the chromosome from becoming frayed or tangled. Each time a cell divides, the telomeres become slightly shorter. Eventually, they become so short that the cell can no longer divide successfully and the cell dies. So, if this is your chromosome, telomeres are present here. Okay, here your telomeres are present. So, they are present at the end of the chromosome. Telomeres are present at the end of the chromosomes. And each round of successive replication makes the telomeres even shorter in length. So basically what happens when your replication happens, the telomeres with each round of cell division becomes even more shorter and shorter. And it in a way protects the genetic material from getting lost. Just imagine if there were no telomeres, then what would have happened? Then, in spite of telomeres getting shortened, your important genetic material would get shortened with each round of cell division. But it doesn't happen because to protect the genetic material, we have telomeres at the end. Okay? And when the telomeres becomes so short that with another round of replication, there is a possibility of losing our genetic material. At that time, the cell can no longer divide and then the cell dies. So cell has a life. So this the life of the cell also depends upon the telomeres. So cell is viable up till we have telomeres of desirable length. After the desirable length of telomeres is reached, then the cells cannot divide further because further division can lead to a loss of cell's genetic material and therefore the cell dies. So let us see, like I was explaining you, if this is your chromosome, this is the centromere, this is the short arm, this is the long arm, and at the ends you have the telomeres. Okay, so what is a telomere? It is actually 5 to 8 base pair G-rich tandem repeats, and basically these are repetitive non-coding DNA. Telo means end, mere means body. That means at the end which is present, and these are non-coding DNA. Important is that these repeats are non-coding DNA. Why do we have them and what telomeres do? So first of all, let me just see so that you see. Now the question comes that what do telomeres do? So what is the function of telomeres? So the first function is that they protect the chromosome, obviously because they are present at the end of the chromosomes. So they protect the chromosome from any kind of nuclease activity, and also they protect the chromosomes from getting degraded with each successive round of cell replication. They separate one chromosome from another in the DNA sequence. Okay. Then, without telomere, the ends of the chromosome would be repaired, leading to chromosome fusion and massive genomic instability. So, if there are no chromosomes, then there can be genomic instability. So, therefore, if there are no telomeres, there can be massive genomic instability, and therefore, telomeres are very much essential. So, telomeres are also thought to be the clock that regulates how many times an individual can divide. Telomere sequence shorten each time the DNA replicates. So, what they are trying to say is that this is your DNA, okay? And at the end you have the chromosome or telomeres. So it is actually the clock that regulates how many times an individual cell can divide. An individual cell can divide only up till the time it has sustainable amount of telomeres at the end. If the telomeres are not present, then what will happen? Then the cell will be unable to divide, because then there is a chance of the loss of the important genetic material. So telomeres decides that how many times a cell can divide. Okay. Then we come to a very important concept that we are going to deal in today's class, and that is the end replication problem. What is the meaning of end replication problem? The DNA at the very end of the chromosome cannot be fully copied in each round of replication, resulting in a slow and gradual shortening of the chromosome. So, to prevent this gradual shortening of the chromosome, we have telomeres. Because what happens at each round of replication, the up till the very end it cannot be copied, so some part of the telomere is only lost, you understand, because the DNA cannot be copied up till the very end of the chromosome. So now, because we have telomeres. So the very end of the chromosome is now telomeres. They're not the actual genetic material. So with each round of successive replication, there is a shortening of the chromosome, but that chromosome shortening is not the major part, but actually the telomeres are getting shortened. So there is no loss of genetic information. So telomeres, it protects. What has happened? It the loss of genetic information. Then we come to telomerase. Now telomerase is an enzyme, okay, which add the telomeres on the ends, which which adds the telomeres. So telomerase is an enzyme that is capable of renewing or adding the telomeres or telomeric repeat onto a chromosome. Okay. What is telomerase? Telomerase is an RNA protein hybrid that has an internal RNA template and a protein outside. Telomerase is nothing but a reverse transcriptase with a single stranded phenotype, as it adds telomeres onto the DNA strand only. Important is that telomerase is basically a reverse transcriptase. Okay, this is important point. Always remember that telomerase is a reverse transcriptase. And what is the sequence of telomerase? TTA GGG. It is important. You should remember the sequence of the telomeric repeat. This is important. Now, Dr. Jerry Shea and his colleagues find out that cellular aging can be bypassed or put on hold by the introduction of catalytic component of telomerase. So what is the meaning? That cellular aging, the death of the cell, is happening because telomeres are getting shortened by each round of cell division. And as telomeres get shortened, that means the cell has to die, because then what will happen? Then, just a minute. Hmm. So then what will happen? Because if there are no telomeres, then the cell has to die after some time. Why the cell has to die after some time? Because then there can be a loss of genetic information, which can lead to genomic instability. So that means if telomeres are getting shortened by each round of cell division, the cell eventually dies. But what if there is a mechanism by which telomeres are getting renewed and replenished, and there is no loss of telomeres at all? So how this can happen? This can happen when telomerase enzyme is always active, and this telomerase enzyme is in a way always renewing the telomeres, and therefore the cell never dies. So what is written over here? That cellular aging can be bypassed or at least put on hold. At least we can see, what we can do is, we cannot make any cell immortal. We should not make any cell immortal also. So what we can do is that instead of making it immortal, we are just extending its period, life period. We are just extending its life period. So now, if it could live for X number of days, it can live for X plus N number of days. So we are just extending its period. So that can also be done. So that is what is a thing. So for that, you need not to add the whole telomerase enzyme. Even if you are adding just the catalytic component of telomerase enzyme, even that much is sufficient for bypassing the cellular aging or putting it on hold. Okay, I hope this point is clear to everybody. Next is, so now what is a telomerase? Telomerase is an enzyme. I told you telomerase is a ribonucleoprotein complex that has been referred to as cellular immortalizing enzyme. Why it has been referred to as cellular immortalizing enzyme? Because it is making the cell immortal. By adding the telomeres, by replenishing the telomeres, it is not allowing the cell to die. And since it is not allowing the cells to die, it is making the cell immortal. Then it stabilizes telomere length by adding the hexameric TTA-GGG repeat onto the telomere end of the chromosome, thus compensating for the erosion of telomere that occurs in its absence. Telomeres activity is observed in fetal tissue, adult germ cells, and in tumor cells. This is an important. Why is it an important point? Because you should remember that not all the cells show the property of renewal of the telomeres. Okay. Cancerous cells have a very enhanced telomerase activity. The activity is also observed in germ cells, in fetal tissues. And activity is nearly undetectable in somatic cells. This is again important. The activity of telomeres is nearly undetectable in somatic cells. Again, important point. Okay. Somatic cells does not show telomeric activity. Somatic cells, they die, but your tumor cells, they have a very profound telomerase activity. Your germ cells, they also have telomeric activity. So all these cells have telomeric activity. Now let us see how does telomerase work. So research also shows that the counter that controls the wasting away of telomere can be turned on and turned off. The control button appears to be on telomerase, which can rejuvenate the telomeres and allow the cells to divide endlessly. Most cells of the body contain telomerase, but it is in off position, so the cell is mortal and eventually die. So basically, telomerase, the activity of the telomerase is turned on or turned off. Okay, this you have to understand. If the activity of the telomerase is turned off, which actually happens in most of the cells, so the activity of the telomerase is mostly off in the somatic cells, and therefore the telomerase ultimately dies after some replications. Whereas some cells of the body, here the telomerase in the on position. What are those cells? What are those cells where telomerase is in on position? Tell me. So mostly in the germ cells, in the fetal tissue. See, these are the things: fetal tissue, adult germ cells, and tumor cells. So in these places, the activity of the telomerase enzyme is on. So how does all this link together? In the laboratory, cells in tissue culture with introduced telomerase have extended the length of their telomeres. Okay. They have already divided for 250 generations past the time they normally would stop dividing and are continuing to divide normally, giving rise to normal cells with the normal number of chromosomes. Okay. So now let us see that how these telomeres actually work. Okay, so telomerase works by adding back the telomeric DNA to the end of the chromosomes. Obviously, it's an enzyme. So what will it do? It will add that ATT-GCC unit, a repetitive unit at the ends of the chromosome, thus compensating for the loss of telomeres that normally occurs as cells divide. Most normal cells do not have this enzyme, as thus they lose telomeres with each division. So what if all the cells of the body has this enzyme? Would have this enzyme, then what would happen? If all the cells of the body would have this enzyme, then none of the cell would actually die. All the cells have telomeres. So what will happen? They will continue to grow. They will continue to add telomeres. The cell will continue to divide. The cell will never die. But this doesn't happen. So in humans, telomeres is active in germ cells. In vitro immortalization, okay. The vast majority of cancer cells and possibly in some stem cells, okay. In vitro, if you are making any cell immortal, then the telomere would be active. In vitro, I'm talking about. Or you have any tumorous cells. In tumorous cells also, the telomerase activity is very much there. And maybe in some stem cells, the telomerase activity is there. Other than that, no. High telomerase activity exists in germ cells, stem cells, epidermal skin cells, follicular hair cells, and cancer cells. Cells which continuously needs to regrow. See, this is important. God. Exist in germ cells, stem cells, epidermal cells, follicular hair cells, and cancer cells. Okay. Then some cells are immortal because their telomerase is switched on. See, telomerase, if it is switched on, that means if it is active, then the cell becomes immortal because continuously it is adding the telomeres. Examples of immortal cells are blood cells and cancer cells. Cancer cells do not age because they produce telomeres, which keeps the telomere intact. So that is why in cancerous cells, you see that there is uncontrolled proliferation of the cells. Why uncontrolled proliferation of the cells is happening? Uncontrolled proliferation of the cells is happening because the telomeres are continuously dividing. Since telomeres are continuously dividing, since telomerase is continuously adding the telomeres, there is no problem at all. Okay, so that is what happens. So the telomerase control gene has been mapped to 3p21, this chromosome. And although the genes for telomerase is present in all cells, HTRT is present only in immortal cells, where it serves to fuse the repeating sequences of DNA into the chromosome, thereby lengthening the telomeres. Okay, that is fine. Now comes important topic, which is telomerase and aging. What is telomerase and aging? Healthy human cells are mortal because they can divide only a finite number of times, growing older each time they divide. Thus, cells in an elderly person are much older than an infant. So basically, why the cells in healthy human, what happens that the cells are mortal because they divide and then they grow old and then they die. Therefore, if you see the cells of an elderly person, they are older and then the cells of an infant. So the discovery of the function of telomeres and telomerase is the single most important discovery in the field of anti-aging medicine. It is a very important discovery in the field of anti-aging medicines because it helps the cells not to age. Fine. So now let us see that telomerase helps in extending the age. So what causes aging? How the aging is actually caused? So there is a term which is known as Hayflick number. What is the term? Hayflick number. So what is this Hayflick number? Cell division is a finite biological process and it is inexorably tied to the aging process. How? Hayflick number describes the number of times a cell can divide. This is important. Hayflick number describes the number of times a cell can divide. Okay. Hayflick number was the first to suggest the relationship between cell division and mortality, but the mechanism was not initially understood. So, how many times the cell can divide? That is your Hayflick number. So, how many times a cell can divide is dependent upon what? It is dependent upon, now we know that it is dependent upon telomeres. Earlier it was not known. So here it is, they are saying that Hayflick was the first to suggest the relationship between cell division and mortality. But how it was related, it didn't knew at that time. But now we know that cell division and mortality is dependent, and this Hayflick number, they all are connected with the telomeres and telomeres concept. How? Because if the telomeres are getting shortened by each round of cell division, there would be a point when the string of telomeres will become so shortened. If the cell will divide, there can be a loss of genomic content, and therefore it can lead to genomic instability. So therefore that cells will not divide further and the cell will die. However, if the telomerase enzyme is turned on, it is very much expressed, then what will happen? Then the cell will keep on dividing and cell will not age. So the aging of the cell also depends upon telomeres. If the telomeres are getting shortened and shortened and shortened, then aging will not happen. But when aging happens, which means the telomeres can no longer are getting uh, telomeres are no longer getting shortened; they are getting replenished with the help of telomerase enzyme. So now comes the end replication problem that we have discussed. That with each round of cell division, what happens? If this is your chromosome and this is the end, the replication cannot happen up till the end of the chromosome. So each round of cell division will lead to the loss of some part of the chromosomes. But is it good? Is it required that with each round of cell division, the chromosome should get lost? No, because we need the chromosome and we need all the genetic content. So therefore, what happens that with each round of cell division, the chromosome should not get the genomic content of the uh, cell should not get compromised. Therefore, nature has installed a mechanism that we have telomeres at the end. So even if the replication is not happening completely, the thing which is getting lost is just the telomeres, which are non-repetitive, non-coding repetitive sequences. So there is not much problem in losing the telomeres. So this end replication problem is solved with the help of telomeres and telomerase enzyme. So let us see what is written over here. That DNA polymerase can only synthesize DNA in 5' to 3' direction, resulting in discontinuous replication of the lagging strand. We all know that there is a leading strand in replication and there is a lagging strand in replication. The replication is continuous in the leading strand, whereas lagging strand with the replication is discontinuous, and we have Okazaki fragments in that case. Consequently, DNA synthesis do not extend to the very end of the lagging strand. Now, it is important. Okay. See, DNA synthesis. You just mark it. I'm very bad at these things, so just pardon me. DNA synthesis does not extend to the very end of the lagging strand. In the absence of a protective mechanism, what will happen? The end replication problem means vital genetic material would be lost during each cell division. So each cell division will lead to a loss of the important genetic material. So telomeres solve this end replication problem by protecting the cell from the loss of critical coding base pairs. This is what we were discussing. Okay. So now comes telomere length as marker of aging. So now with all the information we have, can you tell me that how telomere length be a mark of aging? How telomere length can be a mark of aging with all the information that we have right now? Think. How can we do that? Okay. See, if the telomere length is very good, that means the cell is very new. If we have very shortened telomeres, that means that the cell has aged. And because it has undergone so many rounds of replication, that the telomeres have now become very much shortened. There is an age-dependent attrition of telomere length with losses ranging from between 30 to 150 nucleotide pairs per replication depending on cell type. Cell division or chromosome shortening continues until a critical telomere length is reached, at which point the cell is forced into senescence and no longer replicate. This is what I've been telling you since the starting of the class, that cell division, with each cell division there is chromosome shortening that is happening. So this cell division and ultimately chromosome shortening will keep on happening until a critical length of the chromosome is telomere is reached, and now the cell decides that okay, now after this the cells cannot replicate, because then there would be no telomere left and there would be only loss of the genomic content. And that can lead to what? It can lead to, genomic instability. Therefore, at this point, the cell is forced into senescence and it can no longer replicate. Cellular senescence prevents the replication of incomplete or damaged DNA. This is an important point. Okay. What is the important point? That cellular senescence prevents the replication of incomplete or damaged DNA. Now, telomeres tell how old they are. How? Just read this line. Read this line. Telomeric mediation of gene expression may explain the relationship between telomere length and aging. That means that if telomeres are there, that means if the telomeres are of substantial length, that means that a cell is not aged well. If the telomeres are very short, that means that the cell has aged properly. Now, telomeres may affect all four structural levels. What is the meaning of this? Primary and secondary structure. Telomeres protect the integrity of the base pairs and preserve the basic genetic information. Yes, that is okay, because it is helping the DNA to replicate while preserving the genomic content, and therefore it preserves the basic genetic information. So primary and secondary structure is completely understood. Let us see how it affects the tertiary and the quaternary structural level. As telomeres shorten, they may exert different steric and electronic effects, resulting in the changes in the shape of DNA. These changes may in turn affect the genes which are exposed and available for transcription. That means that the shape of the DNA can be altered because of telomeres. So, if the shape of the DNA is altered because of telomeres, then what happens? That the genes which are exposed or genes which are available for transcription, that can also be altered. So, this is protected with the help of telomeres. So, that is why telomeres are important at all the four structural levels, be it primary, secondary, tertiary, or quaternary. At all the structural levels, telomeres function is important. Now we come to what do they do. Just quickly read this point in 2 minutes and then we'll discuss it. So telomeres are more than a genetic clock. So more than a genetic clock. Genetic clock means that telomeres not only decides it up to how much time a cell will live, the how much time a cell will survive. So for how much time a cell will survive, that is a genetic clock. But it is not just a genetic clock. It is a protective cap. Like it protects the chromosome from recombination, exonuclease degradation, and end-to-end fusion. It distinguishes telomeres from DNA double-stranded breaks, and it prevents the oncogenesis. So all these things are happening with the help of telomeres. Now, just importance of telomeric proteins. I want you to quickly read all the points, and then we'll just this is nothing very much to explain here. The main concept about telomeres we have already explained. This is just like conceptual information. I mean, just an information that you can just read it out. Okay, so just read it. So, what are the importance of telomeric protein? It helps in double-stranded break repair. Non-homologous end joining and homologous recombination have been shown to play a role in survival and alternative telomere maintenance in cells that are detected for proteins involved in telomeric maintenance, elongation, and protection. So these are all the points. Then nucleoprotein complex, number of different proteins that binds to the telomeres, and this is all what you can just read it out. So I'm just marking read. Then this is the genetic clock. See here, this is the telomere length and this is the cell replication. So as the cell continues to replicate, the telomere length keeps on decreasing in the case of somatic cells. These are somatic cells. See here, this is the telomere length and this is the cell replication problem. Cell replication. So you see that the germ layers and the stem cells, even though cell replication is happening, there is not much decrease in the length of the telomere. Why? Because the telomerase enzyme is very much active. Whereas in the case of somatic cells, the telomerase enzyme is not that much active, and therefore, what happens that the length or after each round of cell division, the length of the telomere keeps on reducing. But if there is an by because of some any kind of induction can lead to the activation of telomeres, then if there is a formation of tumor cells, then in tumor cells there is a highly prolonged activity of telomerase enzyme. So that is one thing. So this is the genetic clock. So telomere length was assessed in 143 normal unrelated men and women. Individual with shortest telomeres had significant survival rates, decreased survival rates. Okay. Now we come to C. Relationship between telomere length and age-related conditions. Just look at this graph and try to analyze them. So what you can see over here, that with disease and without disease, hypertension without disease, you see that, and with disease you see that there is a decrease in the length of the telomeres when they are in diseased condition, and without disease condition the telomere length is high. So telomere repeats are reducing, then chances of MIR also increases. Here also you can see over here. Then comes the genetic clock. Now this is an just try to see this figure for 2 minutes and then I will explain. See here, germ cells, cell division and telomere length. What is happening? With each round of cell division, the telomere length is constant. In the case of germ cells, it is constant. But in somatic cells, the telomere length is keep on reducing, and there is a Hayflick limit. Hayflick limit. So Hayflick limit is reached at a stage of M1 senescence, that is the first initiation phase of senescence. Division continues with lots of difficulty and it reaches the M2 phase, that is called the crisis phase. So M1 phase is the senescence phase and M2 phase is the crisis phase. Now crisis phase end up having genetic instability. So if the cells goes up to the crisis phase, that means it will lead to genetic instability. Germ cells maintain long telomeres indefinitely. The line is horizontal, you can see, indicating no telomere shortening occur due to telomerase activity, whereas in the case of somatic cells, it shows a steady decline in telomere length with each division due to lack of sufficient telomeric activity. So genetic clock is an interrelationship between the length of telomere and the number of times a cell can divide further. Okay. So, situation in which the cancer is progressed. So, these are the various situations in which the cells can progress the cancer. What is the situation? First situation is it can bypass cell cycle arrest checkpoint pathway. Then the tumor suppressor genes, P53, which is also known as tumor protease, a transcription factor that regulates the cell cycle and functions as a tumor suppressor. And RB is the retinoblastoma. It is critical for cell cycle exit when dividing retinal progenitors. So these are the tumor suppressor genes are of this. If they are not expressed, then continue to divide very short telomeres. No longer it protect the ends. Cells enter secondary proliferative block called crisis. So these are situations in which the cancer is progressed. Just a minute. Okay, fine. Mm. So now comes again the same thing that we are discussing, the genetic clock. The telomere length, as it comes down, it will reach a Hayflick limit. This is the Hayflick limit, you can see. That is the limit in which the cell can no longer divide. So Hayflick limit decides that the cells can no longer divide. Okay. So when I talk about here, cells can no longer divide. Until a critical length of chromosome, the reduced telomeres are tolerated. Okay, now here the cells can no longer divide, but until even that, until you can tolerate, the cells can tolerate the reduced length of chromosome. But once it goes, that it will eventually lead to a crisis phase. In crisis phase, the cell will completely stop dividing unless it gets a second chance in the form of cancer. So the telomerase activity has to be turned on. So what happens in the crisis phase, but because of some kind of external factor, what happens if the telomerase activity is turned on? Then at that point, what happens? The cell become immortalized. The cells become immortalized means that now it will grow, it will again indefinitely keep on dividing because telomerase will keep on adding the telomere repeats. Okay. So summary, replication problem. Therefore, there are telomeres and telomerase, and telomere dysfunction can lead to cancer. Okay. Maintenance and manipulation of telomere length. So factors that accelerate telomere shortening, it is all not important. Now, these are some of the examples. Okay, I'm writing here examples. These are the examples that tells that what are the factors that can accelerate telomere shortening. So you see that there are some factors that can accelerate telomere shortening. What are those? Yeah. So stress can lead to the shortening of the telomere. So there are certain factors that can accelerate the telomere shortening. Okay. The first factor is stress. Then there is vitamin C. Then there is omega 3. It can slow down the telomere shortening. So omega 3, it can, what this happens? Mm, It can, then telomeres can restore the youthful phenotype because telomeres can add the telomeres, and telomeres are added, the cell will not age that much. Now we come to end questions. So the first question is, end replication problem is elevated by RNA polymerase, telomeres, reverse transcriptase, or telomerase. So what is the correct answer? End replication problem is elevated by what? So after 2 minutes, I will switch the screen. Think what can be the answer? What can be the answer? Think. One more minute left. Okay, time's up. So the answer is telomeres. So end replication problem can be solved by telomeres. So if you are adding telomeres, then what will happen? The important genetic material is preserved. And when the important genetic material is preserved, then what happens? The end replication problem is solved. Now the next question is, which of the following causes chromosome compaction? snRNA, PRC2, shRNA, and HAT. Think and try to answer. Think. What can be the answer? So the correct answer is PRC2. How it is the correct answer? Think. PRC2 basically causes the chromosome compaction. So PRC2 adds the methyl group to histone H3 at the lysine 27, and this repressive histone mark leads to chromatin compaction and gene silencing. Okay. What happens? That this PRC2, it adds the methyl group, and when it adds the methyl group, it causes the methylation. And since it causes the methylation, it leads to chromosome compaction, and therefore it is involved in epigenetic regulation and developmental gene regulation. So it is also involved in epigenetic regulation. Epigenetic regulation. Now the next question is, telomeres can be visualized using real-time PCR, Nathan blotting, fluorescence microscopy, or Western blotting. Which technique can be used to visualize the telomeres? Think. Think. You have 2 minutes to answer this question. Which technique can be used to visualize telomeres? Real-time PCR, Nathan plotting, fluorescence microscopy, or Western blotting? Think. So, what is the correct answer? The correct answer is fluorescence microscopy. How fluorescence microscopy is the correct answer? Because telomeres, it can be visualized with the help of phase fluorescence in situ hybridization. What it will do, a fluorescent probe that binds specifically to telomere DNA repeats, which allows direct visualization of telomere length and location on the chromosome. If it is real-time PCR, it can quantify the telomere length, but it cannot visualize them. Nathan plotting में you can detect RNA and telomeres are DNA, so you cannot detect it. Western blotting में again it is for protein and telomeres are DNA. So this is for RNA. This is for protein. Here in real-time PCR, what you can do? You can quantify telomere length, but visualization, no. So only fluorescence microscopy can be used to visualize the telomeres. Okay. So the next question is Hayflick number. Hayflick number refers to age of a cell, stage of DNA replication, number of times a cell can divide, or cellular senescence. What is Hayflick number describes? Think and try to answer this question. Right now it is 4:58. I will switch the screen after 3 minutes. Okay. Think. One more minute left. Okay. So the answer is number of times a cell can divide. So we have discovered Hayflick number refers to age of a cell, stages of cell replication, number of times a cell divide, and cellular senescence. So Hayflick number refers to the number of times a cell can divide. Okay. So Hayflick number will decide that what is the length of the telomeres and how much time the telomeres can help in the replication, and then how much time the cells can divide. Okay. The next question is telomerase can come under the enzyme category of reverse transcriptase, nuclease, DNA polymerase, and DNA ligase. So, which is the category under which telomerase come? It comes under the category of reverse transcriptase. It comes under the category of nuclease. It comes under the category of DNA polymerase, or it comes under the category of DNA ligase. Think right now it is 5:03. I will switch the screen after 3 minutes. Think. Okay. So the correct answer is reverse transcriptase. So we have already discussed that telomerase is basically a reverse transcriptase which takes the RNA template and adds the DNA subunits over there. Okay. The next question is cancer risk is high when telomerase is inactive, telomerase is short, P53 is active, or telomerase is active. So when is the risk of cancer is high? think what can be the correct answer? Cancer risk is high when telomerase is inactive, telomerase is short, P53 is active, yeah, telomerase is active. 1 more minute left. The correct answer is telomerase is active. So, cancer risk is high when telomerase is inactive, telomerase is short, P53 is active, and telomerase is active. So, obviously, when telomerase is active, what will happen? It will keep on adding the uh, telomeres, and when it keeps on adding the telomeres, what will happen? Then the cells will not die. And when the cells will not die, what will happen? Cells will become immortal. And when the cells will become immortal, then what will happen? It can lead to the risk of cancer. So, cancer risk is high when telomerase is active. Now we come to the true and false. Telomere is a repetitive sequence present at centromere. True or false? Tell me. You have 3 minutes to answer this question. Telomere is a repetitive sequence present in centromere. True or false? Okay. So let us see what is the correct answer. The correct answer is false. That is, telomere. Is it telomere? Is a repetitive sequence present at the chromosome? No. At the centromere? No. Why? Because telomere is present at the ends. That is why we're talking about end replication problem. Na? If we're talking about end replication problem, that is because we're talking that telomeres are present at the ends. Then only they can solve the end replication problem, and then only they can help in preventing the chromosome shortening. So telomere is a repetitive sequence present at the centromere. False, because it is present at the ends. Next question is, cellular senescence is seen in all cell types. True or false? Think. Okay. So the correct answer is false. Why it is false? Why cellular senescence is not seen in all cell types? Because we have already studied that cellular senescence is seen only in germ cells, few of the stem cells, in tumorous cells. But in cellular senescence, cellular senescence is seen in somatic cells. But tumorous cells, do they show cellular senescence? No, they do not show cellular senescence because they have infinite activity of the telomerase enzyme. And your germ cells, do they show cellular senescence? No, because they have telomerase enzyme. So cells which show the telomerase activity, where the telomerase activity is switched on, their cellular senescence is not seen. For example, your tumorous cells, your germ cells, and few of your stem cells. Whereas somatic cells, if I talk about the somatic cells, then somatic cells, they do show cellular senescence because there the telomerase activity is off. Telomerase activity is off in those cases. Okay. Next question is, DNA end replication problem is seen only in stem cells. True or false? Tell me, what is the correct answer? DNA end replication problem is seen only in stem cells. So the answer is false. Why? The question is DNA and replication problem is seen only in stem cells. Obviously, it is false. Why it is false? Because DNA and replication problem is seen in somatic cells where telomerase activity is not there. So the cells in which telomerase activity is not there, there you can have end replication problem. But that is also solved because telomeres are there to a certain rounds of cell division. It can go and then the cell will die. So the correct answer would be false. Next question is telomere length is same in all chromosomes. What do you think? It is true or false? Telomere length is same in all chromosomes. Think. So telomere's length is same in all chromosome. What is the correct answer? True or false? Telomere's length is same in all chromosome. The correct answer is false. Why? Why it is false? Do you think telomere length is same in all chromosome? No, because some cells are dividing fast. The cells which are dividing fast, their chromosome length will get shortened quickly. Whereas cells which are dividing slowly, their chromosome length, telomeres length will shorten less quickly. Then cells which are tumorous cells. So tumorous cells and a somatic cells. Will the chromosome telomere length in the tumorous cells or chromosome telomere cell or telomere somatic cell or a germ cell or a tumorous cell would be same? No. Telomere length would be more in a germ cell or a cancerous cell than in the case of a somatic cell. So we can say that telomere length is not same in all chromosomes. Okay. Okay. So the answer is false. So this leads to the end of today's class. I hope you could understand something. And we'll meet in the next class. So next class would be the last class. And in the next class, we'll do the last subject, last class questions, and that would be the end. Okay. So, okay then, we'll stop the class now.